will talk to you a little bit further about her wonderful project called Insect Investigators, which is a citizen science project that she leads, several $1 million of funding. Oh, come on. That's the figure I saw. Doing work in 3 states, Queensland, Western Australia and South Australia. Yeah. So welcome, Erinn. Come up and tell the group a little bit about yourself, what you've been doing since last time you presented, and with her beautiful outfit.
Well, Naomi and I were at Botanic Gardens today. The Botanic Gardens were hosting a World Environment Day for schools. So we were both there running workshops all day. So whenever I work with kids, I've got to wear the bug outfit, dress the theme. Yeah, I'll share it in a sec. I was just getting it open.
We've been doing Zoom for over 2 years now. What will we do without it?
It's just come up.
Bunch of people.
Okay, there we go. Should be right. Okay, hello. So I think the last time I talked to you guys, I think was 2017, 2018 maybe. I was towards the end of my PhD. So I'm now a postdoc at Adelaide Uni and the South Australian Museum. And today I'm going to talk about this really awesome project that we've been running the last few, well, it sort of started a few years ago, but it's mainly been happening this year. So I work on wasps. Yeah, sure. Sorry, I should have brought a mic. I work on wasps, parasitic wasps. So those of you who have heard me talk before will have heard me talk a little bit about wasps. So I'm only going to talk briefly about them. This is maybe what you imagine when you think of a wasp. This is, of course, a European wasp, invasive into Australia, annoys you at your barbecue, but wasps come in all shapes and sizes. So this is a cuckoo wasp, a native wasp to South Australia. This is a photo by James Dory, who, if you love photos of beautiful native bees, I recommend his book, Bees of Australia. These cool little wasps are ones that lay their eggs inside other bee and wasp nests. So the babies then eat the food provisions that the other mother provided. So they're kind of sneaky, but very beautiful. But I work on a group that looks like this. They're only a few millimeters long, and they're parasitoids of caterpillars. So they lay their eggs inside the live caterpillar, and then the baby wasp, of course, hatches out inside the caterpillar and has fresh, live caterpillar meat to eat. They get bigger and bigger until eventually, of course, they burst out of the caterpillar and look a little bit like this. They make little cocoons, metamorphize, just like caterpillars and butterflies do, into our adult wasps. So this was a sample that came into the Queensland Museum while I was up there visiting a few years ago, and all these little wasps have come out of one caterpillar. So I don't think I would have liked to be that caterpillar that day. That's just an introduction on what I work on. So these are little wasps that I worked on for my PhD and that I work on now for my postdoc. And of course, because nature is crazy, even parasitic wasps have their own parasitoids. So that's just a tiny little hyperparasitoid. So it's looking for any of those little metamorphizing wasps inside the cocoons to lay its own egg on, which is pretty cool. So I work in, I guess, what we call biodiversity documentation or taxonomy and systematics. So taxonomy, as many of you will know, I'm talking to a very sciencey audience here, is, it's about classifying the life around us, naming species, working out how they're related to each other through time. And we think that less than 30% of the species in Australia actually have names. So things like butterflies are relatively well documented because they're quite pretty. Lots of people have worked on them, although even those have lots of probably undescribed species in undersampled areas. But things like wasps and flies, maybe 10% of the species in Australia have a scientific name, let alone us know what it's doing in the environment or whether it could be useful, whether it needs conservation efforts. So there's so much work for taxonomists and there's so few of us and there's so little funding that, we do the best we can with what we've got, but we're really trying to work out ways to accelerate how quickly we can describe species and document what's around us before we start to lose it all. So this project, Insect Investigators, kind of came from the idea of, people don't hear about taxonomy until maybe they're at uni, if they're lucky. Can we get people hearing about taxonomy and being passionate about it and excited about documenting what's around them earlier? So it's about trying to connect that into schools and into school students and raise awareness for what taxonomy was. We also wanted to start building the DNA barcode library for Australian insects. So a DNA barcode is just a little section of the genome that we use to help tell species apart. And there's this real effort to try and document all living things with this little barcode, which can help us know a bit more about things that are maybe morphologically or visually quite hard to tell apart. But the Australian fauna has kind of slowed behind the rate of how we're building that library for the rest of the world. And we also wanted to describe new species, but in collaboration with communities, not kind of hidden away in museums and universities, but actually with the people from the place that species was discovered, so they can feel ownership over it. Well, that's the hope, at least. So the initial trial of this project was a couple of years ago, and I worked with 4 schools in South Australia, so Macclesfield Primary in the hills, Wakery and Ramco Primary Schools, which are in the Riverland, and Howe Area School on the Eyre Peninsula. So I headed out to each of the schools with the help of a little seed grant from the Citizen Science Association. And at each of the schools, I ran a little insect workshop and we set up a malaise trap. So if you haven't seen one before, it kind of looks a bit like a tent in that photo. It's a passive trap, so it doesn't have an attractant to bring bugs in. What happens is the insects fly along, they hit that panel, that's vertical panel down the middle, and then insects will kind of instinctively go up towards the light. They head up to the top and into that collection bottle, which has a preserving liquid in it like ethanol or propylene glycol. You get insect soup, basically, full of lots of flying insects. And you can leave these out for several weeks at a time, and it sort of passively samples the environment around it. So we set one up with each of these schools and then left them with it. They checked the trap, they changed the bottle, and sent the samples in to us at the uni. So there's the trap. This is at Ramco Primary in the Riverland, headed out there with the kids. And this is kind of what the insect soup looks like when we get it back in the lab. So we tip it out in a dish and we start to sort it. So in this project, we sorted to order levels. So say all the wasps and ants and bees, all the beetles over there, and told the kids a little bit about what they'd found. There's all the beautiful wasps caught out at Ramco. And they actually found one of the wasps that I was working on, one of our little microgastrine parasitoids. And actually all 4 schools found a species that had not had a scientific name. So discovered a new species, or new to science at least, of the little subfamily that I work on. And that, I guess, is just an indication of how many things are out there that are just undocumented. So I worked with all 4 of the classes at each of the schools to come up with what they wanted to call it. So up on the top left, that Franklin Harbourensis, which is the council area that Cow's in. They wanted to call it after the area. Dryoplanitis is Greek for bush wanderer, and that's from Wakery. The area where we collected it, they call it bush block. So they thought it was nice to have the wasp kind of wandering around the bush. Ramco marmorata. Ramco is obviously the place. Marmorata is Latin for marbled. They thought the wasp looked like it was made of marble, the black and the white, I guess. And Wakery yetis, Wakery the place. Yetis is their youth environment team's nickname. They're called the Yetis. And it was that group which was leading the project. So they slipped their name in there, even though we talked about the fact we don't normally name species after ourselves. We kind of had an exception in this case. So we worked through taxonomy and how we name species and how we go about that process. And we published the paper. So all 4 of the species were named and published and described formally, which is really cool. So this was kind of what we thought it was a success. We asked the kids what their favorite thing about it was. This is just a word cloud. So the bigger the word, the more commonly it came up in the kids' answers of what their favorite thing was. Most of the answers were finding the new species or finding the new wasp. And so, that discovery element was really their favorite part of the project. Lollies is up there because we ran a workshop where they used lollies to build phylogenetic trees. And then I did the evaluation straight afterwards. So I learned when you run surveys with kids, don't run them straight after you've fed them lollies because that's going to be their favorite part. We asked kids a bunch of questions afterwards about what they felt the program had impacted them or how it had impacted them if it had. So in this case, the question is, did your interest in insects increase or decrease or stay the same? So the blues are increased, the orange is decreased, and grey is it stayed the same. So for lots of kids, they felt the project had meant their interest in insects had increased a lot. Lots of them said their knowledge had increased a lot. Quite a lot of them said their desire to protect the environment had increased, and their connection to nature had increased, and their desire to spend time outdoors had increased. For maybe a third, they said their desire to pursue a scientific career had increased. For a lot of them, it stayed the same. And for some of them, it decreased. So I don't know what I did wrong there. Or maybe we were just doing the kind of science that maybe they're not interested in, or they learned that science takes a really long time, or I don't know. It's kind of an interesting result. But of course, this is just a little survey, and we're hoping to do some better evaluation on this project. So that was a little trial. And it launched kind of the second iteration. So we brought together some really cool partners, which you can see all the logos there, to apply for one of the Australian government citizen science grants. And we were very, very lucky to be successful. So we got $500,000 to run a much bigger version of this, which was really exciting. So basically, again, we're going to get schools to set up a Malays trap. They will collect insects. We'll do some DNA barcoding, so sequencing that little bit of the genome that's really useful for telling species apart. We were then going to get the specimens and send them out to taxonomists or experts in different groups, hopefully find and describe some new species and get that published, all within the space of 2 years. So it was a bit of a rush job. But we pulled it all together, we sent out the applications, and we got over 200 schools apply to be part of the project. So that was kind of interesting to me and super exciting. I wasn't sure that teachers would have the time or the capacity, especially this year, to sort of do extra stuff like this. But it almost seems like there was more interest because it's been such a hard couple of years. And I don't know if you found that with the butterfly stuff, but I just thought teachers would be so pushed with everything else, they wouldn't want to do anything extra. But we got heaps of interest, which was awesome. So we only had 50 traps. That's what the budget was for. So we had to select 50 schools. We tried to get kind of a range of geographic locations and socioeconomic areas. And this was our end result. So these are the 50 participating schools in WA, South Australia, and Queensland. So some really cool schools. We've got Mornington Island up the top, Manili near the Northern Territory border, some up in the Kimberleys, some right across the west, so that's Yalata, west of South Australia. Yeah, some really, really interesting schools. We're so excited about the insects they've collected. So we made some resource videos. We made a teacher resource with some lesson plans that were kind of generally about insects and taxonomy. This is a little video we made about how to collect bugs and how to set up a Malays trap with a couple of interns at the South Australian Museum, Samika and Emily, who are part of the Aboriginal science internships they do there, which is really cool. We had to send all the schools all the equipment they needed. So inside all of these boxes is one of those traps that you saw the picture of. They got a net to do a bit of collecting themselves of insects, a bunch of vials to put things in, and a few other bits and pieces, some hand lenses and magnifying glasses and things like that. And all of them made it to the schools. You can see in the top left, there's some of them got a little bit beaten up in the post. That one was going out to far out regional Queensland, but they all got there, which was really exciting. We then tried to actually visit the schools and run a bit of a workshop before they had to set their trap up. So this is my collaborator, Andy Howe, up in Queensland. On the left there, there's a school way out there in the distance called Cameron Downs, basically in the middle of nowhere. He did about 5,000 Ks visiting the 16 or 17 schools in Queensland, running some workshops and things. In Western Australia, they got to about half of their schools. That tiny little plane they caught out to Shark Bay to go and visit the school there. And this is Barbican Primary School on the right-hand side there, and that's the entire school out collecting bugs for the day. So, you know, these kids don't often get visits from scientists or from educators or, you know, anyone outside the community. So it was pretty exciting that we got the chance to head out there and see them. Unfortunately, in South Australia earlier this year, Education Department wasn't allowing incursions, so we did all of our visits by Zoom. There's me and Sylvia and Alice from the Murray and Riverlands Landscape Board, who are our partners in South Australia. And hopefully we'll visit the SA schools later in the year with some results. So all of our schools popped up their traps and they ran them for 4 weeks in March. So here's just a few of the different environments. That's Kangaroo Island up on the left. I think this one is Trinity Colleges. Can't remember if I recognize the uniform. This Yalata, Tambourine Mountain. Well, maybe that's Trinity's with the fence. One of the South Australian ones sent me this picture with their Malays trap in this little fence, which I thought was super cute. Apparently it's to stop the kangaroos bumping into it. I'd never seen that before, but good invention. So yeah, lots of really cool locations, really diverse habitats. And then of course, with all the other resources, we encourage them to do a bit of catch and release collecting at the same time. So one of the schools, Elliston, made their own little pooters or aspirators. So you suck up the bugs through one end and it goes into the vial, which I thought was really cool. And, you know, get out and catch bugs, use iNaturalist, take photos, see what's around you, and then be able to compare that to what you're catching in the trap. These kids are super cute. Little reception year ones. We had reception year ones right up to high schools involved. So a really big mix. There's the insect soup that got sent into us. And then we had to basically process. So each school ran the trap for 4 weeks. So we had 200 bottles coming into us here at the uni. So we got super excited when the first couple of packages arrived, express post, with our little trap sample in it. And then by about the 100th, the novelty wears off because you're like, oh gosh, now we have to process all of these insects. But basically we unpack them. We are taking off a little sample of the liquid to see if we can do some cool genetics down the line, some eDNA work. And we're measuring the biomass of the first 2 weeks and then sort them to order. So we had this awesome team of volunteers, well, no, we paid them, but of students doing casual work for us to sort all these traps to order level. And we couldn't have done this project without all that kind of man time and labor from them. So it was amazing to have them involved. Here's just a few examples of what they look like. So this really soggy one down in the bottom right is Cameron Downs. They had a grasshopper explosion while it was on. So it was basically just flooded with grasshoppers. I think that's Elliston in the middle at the top. And on the left is Kalumburu, which is right at the top of Western Australia. So real diversity. You guys all know what the butterflies are. Maybe? No, I don't know. I'm like, it's a butterfly. My knowledge doesn't go that far. So the idea was we got the traps in, we unpacked them. So the school sent us the bottles that had their sort of insect soup in it. We kept them cold until we needed to process them. So this was sort of our fridge full of insect soups. We then started to have a look what's inside. So every trap we take a little picture, which we're hoping to be able to, do some comparisons down the line. And then our team sorted them to order. So each of those 200 bottles got sorted into some nice little files, each with different sorts of insects in it. So that's kind of the sped up process. We go from insect soup to vials of insects sorted to order. I don't know how to turn down the volume on this, but this one's going to have music as well, so at least you know what it's like. What it's all about. Check it out. Check it out. Check it out. Let me show you what it's all about. Just a few of the bugs we found. So Malaise traps are mainly used for catching flying insects. So the red colours here are lots of numbers of insects. Yellow is a lower number of insects. Those two big red lines are the number of flies and the number of wasps and bees that were sent off for each of the schools, which are along the x-axis there. So all of the schools, we sent off over 100 specimens of wasps and flies, and then lots of other kinds of insects as well. And when I say sent off, what we did was collect out a bunch of insects that we had experts particularly interested in working on, and we've sent them actually to Canada, where their DNA Barcode of Life institution works, and they're going to process those samples, sequence that little bit of the genome that will hopefully tell us a bit more about it, and send the specimens back. And we'll then send them out to taxonomists who are experts in different areas. So some of you might know Ned Fisher. He's a moth guy in Adelaide. Very passionate moth taxonomist, but still hasn't started his postgraduate studies yet, but he's sort of going to be amazing, I think. And Elise, who's a science student. So this was our 5 boxes of over 14,000 insect specimens that we sent off to Canada, get DNA barcoded and learn a bit more about. So they made it through customs and they're processing them at the moment. So that's kind of where we're up to. We've collected all these specimens, we've sorted them, we've sent them off to get DNA barcoded. We're hoping that will tell us a bit more about them. We're going to get all the specimens back, send them out to taxonomists, and hopefully be able to document some new species. Now, because there are so many insects that are undescribed, the likelihood of finding new species is really, really high. But the chances of there actually being a taxonomist working on that particular group at the moment who can tell us immediately it's a new species is much lower. So there'll be some groups where someone will be able to look at a sample and say, yep, definitely a new species. We can put a scientific name on this and get it published, get it out there into the literature so it can be put on conservation records or used for biocontrol or have more study done on it. But lots of those specimens will simply go into museums where they'll be able to be used by researchers for a long time into the future. So while we're waiting for the results, we're kind of running some scientist webinars. So we're meeting different taxonomists across the country, and they're intended for schools, but anyone can come along. The recordings go up online, doing some blogs, making some little videos for the kids about what we know so far about the insects they've found, and kind of encouraging them to use the lesson plans and get out there and explore the environment around them. So it's a really, really cool, amazing team behind this. Lots of people from all over South Australia, Queensland and WA who are kind of on the ground and in the museums. And then this bottom sort of row and a half are our wonderful taxonomists who have signed on to actually receive these specimens and have a really good quick look at everything and try and get some new species named in collaboration with the schools. So the idea is that if a school can help name a species and feel that that species is theirs and is in the environment, maybe that will help inspire them to want to protect it when they've actually got to name it. Who knows how much impact these programs actually have in the long term, but we're trying to do some evaluation and work out how much impact we do actually have on the kids and whether it does change their opinion on, whether they are more interested in planting native trees or more interested in, looking after that environment that their species is found in. So yeah, that's the project. we're on socials, or there's lots of information up on our website, including recordings of the webinars and lots of kind of documentation about how the project's gone so far. But yeah, we're hoping we get some really good results back and we can get those results out to schools before the end of the year. That's the plan anyway. So yeah, thanks everyone. Any questions? Or any questions on Zoom? Feel free to unmute and ask if you want to.
Yeah, stop sharing.
I guess I should say the other thing is the funding was basically to run it this year. So to do the one collecting, the one processing and get all those results back. And then the project goes to the middle of next year. But the first 6 months of next year, really, we'll just be trying to get these papers published and kind of wrap it all up. So we're currently looking for how we keep this sustainable and how we keep it going and growing, because the feedback we've had from the teachers so far has been really, really great. We've got a discussion board on the website, which is really cute because the kids are posting pictures of what they found when they were doing collecting and talking to each other and all that kind of stuff. So check that out if you want to feel some warm fuzzies for 5 minutes, because it's really nice just to actually see them in action. But yeah, how we actually keep it growing and keep it going. So if anyone knows awesome funding sources, you probably struggle with this too, how you keep things going, let me know.
Yeah. Yes, Gil. We'll repeat the question.
Did you start this off yourself?
Yeah, so Gil asked whether I started it off myself.
That's fantastic.
Yeah, the trial was me, but this, like the bigger version, obviously, is not just me. It's heaps of wonderful people.
I've always wondered, as wildlife conservation, concentrating on butterflies. Most of them said, well, it's not getting attention.
No, but I think butterflies help bring people in. Yeah.
I wish I was younger joining that group. I really love it.
So Gil was just commenting on the fact that, you know, we've done a lot of work on butterfly conservation, and then recently a substantial work on moths, and yet more work to come with the Caterpillar app and the smartphones and things like that. So this is why collaboration is so important. And, you know, Erinn hasn't told you, but she's got a master's in science communication. So it really shows that, you know, you connect with people, you connect with nature, and you get the job done. Gary.
I wonder again about a strong connection with both council and wetland areas in each council area. If there's no visible indication of the insect life in the wetland area, it is something that's the essence.
Yeah, it's a very interesting observation Gary's just made about the fact that in many of the wetlands and other parks, we hardly ever see any documentation of the insect life that's in there. And one case in point, the little gums reserve that I'm working in at the moment, we had David Hurst came and did a spider walk with us. And we're about to do some rehabilitation on the creek. And I said to the council, we need to do a fauna survey. Even though it's a degraded site, there is still insect life there. And so, you know, it'll be no good if we dig up that bank and lose those trapdoor spiders that he found with us, because they're gone once they're dug up. Yeah, so certainly having some insect education. So, you know, I like you, are members of SA Nature Tears on Facebook, and we know that there are lots of experts out there doing observations in those places.
Certain sites out there, so identify pictures of this insect. I wonder what it is, but it's good sharing.
And I think iNaturalist is amazing for that, because you have a wasp expert who maybe lives in Canada but is able to identify pictures of wasps from Australia because they're uploaded on that site. So yeah, amazing chance to kind of get that documentation happening more broadly.
Atlas of Living Australia.
Yeah.
Yeah.
Another question from the floor, and then we'll ask the Zoom audience. Yeah.
I this isn't really a question, but I thought it was a terribly clever idea as a way of getting thousands of specimens from dozens of places rather than one keen individual going out and making it their life's work to get all those kids. I mean, even if only 10% of them are particularly interested and keen to go on with it, that's magic.
Yeah, and that's one of the awesome things is being able to, like for us, being able to get samples from places that we would never have funding to go do field work is really cool. And that the work's being done by the people who are local and live there, I think is really cool.
And some of those people some of those young people will grab the ball and run with it. They won't lose interest. A lot of them will, because it'll, something else will happen, puberty or something. But a lot of them will.
Yeah, and I think
some of them, the keenest will be the ones who will carry it on.
And I think with a lot of this stuff, it's not necessarily about inspiring all these kids to be scientists, but if they become farmers or teachers or, exactly.
And to realise that if you clean out all the insects, what are the birds going to eat?
Yeah.
And if you clean out all the insects and the birds, then what? I think the whole idea of, in a way, it's good that you're doing insects, but it's because they're not fluffy and they don't have large round eyes. But, well, some of them do, but they're not as visible as the large round eyes on koalas. But the idea that nature, I think there's a lot of a fixation upon what mammals are doing. And if you're not wiping out mammals, go for your life with your bulldozer. And I think it's very, very wrong. It's always been wrong, but it's good to get it out there that the whole thing is interlinked, interwoven, and intimately involved with different species, the different environments, the different minerals in the soil, the different life forms, vegetable or animal. But the whole idea that you can just have, nice koalas sitting in the right sort of gum trees, and so everything's cool and sweet, is unfortunately still rather prevalent.
Not indigenous itself.
Oh, but you could say that till you were blue in the face, but aren't they lovely? I mean, I'm not anti-koala, but I'm not obsessed with the bloody things. And when one of them gets bowled on the road, I probably don't get as upset as I ought to. But I hate to see bulldozers. I mean, look, there's a little site across the tram line from us, which means it's in Marion, where there was a lot of housing trust places, a lot of not very well off people living there. Now, they scraped that site. God knows what was there before they scraped it, but they've scraped it. They've taken the animals, the vegetables, the plants,
all the soil life. Yeah.
They've carted away a lot of the soil, I think. They're covering a great deal of it with concrete. I mean, it's like the backside of the moon. It's dead.
Yeah.
And there's a lot of that sort of development, and they don't look at these places before they go in and disturb every molecule of them.
Have we got any other questions? Any questions from Zoom? No? Well, if not, we'll sincerely thank you, Erinn, for a wonderful talk. It's lovely to have you back. Erinn was a very valuable person to have on our committee a few years ago, and we really value your opportunity to come and speak to us on the great project. So without further ado, I'll give you a copy of our Caterpillars book, and hope you enjoy that, and we look forward to hearing more about the project. So encourage everyone.
Back with results, hopefully, at the end of the year.
Yeah, that'd be great. Encourage everyone to go on that website.